All right, everyone. Good afternoon, Cathedral. This week we are happy to have Dr. Troy Rowan presenting for us. Dr. Rowan is an assistant professor and extension specialist at the University of Tennessee's Knoxville campus. He grew up on a Charolais cattle ranch in Iowa, and he's remained engaged with the beef industry ever since. At Tennessee, his research program uses genomics to understand and predict economically important traits in beef cattle. His group's work is particularly interested in developing novel indicators for cow efficiency, adaptability, as well as mechanisms underlying heterosis. And today he will be presenting on using molecular phenotypes to understand forage-based cow efficiency. So Troy, whenever you're ready, you're welcome to start.
All right, sounds good. Well, I appreciate everybody tuning in today and inviting me, like I said, north of the border via Zoom. So it's always good to interface with your guys' group. I really admire a lot of the work that you do, particularly in the dairy world, and always looking for opportunities to steal some of your good ideas and try and apply them to the messy system that is beef cattle here in the Southeast and across the U.S. So I'm going to sort of walk through three of the big ongoing projects in our lab right now. None of this at the moment is published stuff, so sort of all continually evolving research. But the big thread I think that sort of pulls through all of these components is having to step back, understanding that we're maybe not able to measure directly all of the phenotypes that we're interested in improving in the beef industry. So how do we use novel genomic applications, various types of omic data to get at some of these more complex phenotypes that underlie a cow's ability to forage, sometimes in a stressful environment, and convert that ultimately into weaned calf weight. So again, we're very focused on this commercial cow, and there are a handful of ways that we think that we can address efficiency. And so the big questions in our lab and what I'll talk about today are: how does heterosis work? We'll spend some time on that starting things off. How can we use omics data for phenotyping? So understanding that we sometimes need these latent indicator traits to do useful genetic prediction. How can we use omics and how can we sort of tailor novel genomic approaches to doing phenotyping? And then finally, how can we breed more efficient cows? And lots of moving pieces to this that we'll focus on just a couple. So with that, I want to start off with this story about heterosis. This is a project that a former master's student, Garrett Franklin, he's now working on a PhD at the University of Missouri. Garrett did the vast majority of this work, and it was really an incredible effort, but one that we're really excited about, trying to delve into more mechanistic understandings of how heterosis underlies our complex traits. So everybody likely familiar with heterosis, of course, the overperformance of this F1 offspring compared to its two more inbred parents. We obviously can't generate the same amount of heterosis that our friends in corn breeding can, but we can, to a certain extent, observe this, particularly in our beef cattle populations, many of which are pretty genetically diverged from one another. And so when we're talking about heterosis, what we're actually thinking about is this deviation of an offspring's performance for a given trait compared to the average of their two parents, or that mid-parent value. And our interest, of course, is knowing that heterosis works across traits, but particularly it has an inverse relationship with the heritability of a trait. So for our lowly heritable traits, our maternal ability, reproduction health, of course, lowly heritable, tough for us to make genetic progress on in any rapid way, particularly doing phenotypic selection. But we can see enormous amounts of heterosis in these traits, sometimes upwards of 30% advantages in lifetime cow productivity out of an F1 crossbred female compared with a straight bred commercial female, the average of the two parent breeds, right? So huge amount of evidence that heterosis works. It works across traits, and it particularly is effective at improving these traits that are central to this question of cow efficiency. And so our question that we wanted to dig into here is the evidence of heterosis existing across traits is well established, but there's been very little work trying to understand what the intermediate biology between those non-additive gene effects and an animal's expression of heterosis in a phenotype. Our interest is in trying to understand what happens under the hood, what are the drivers, and can we leverage knowledge of how heterosis works mechanistically to make better, more tailored predictions, particularly thinking about how we might model informative, non-additive variation when we go and try and predict individual animal matings and the heterosis that we can generate from them. So our question is if heterosis happens at the level of the animal, what are the intermediates, right, between those mutations, those non-additive mutations that occur in the DNA, and the ultimate phenotype, right? Understanding that the central dogma of biology tells us those mutations are manifest in differences in transcription, and ultimately translation, which is going to affect that end phenotype of an animal. So our focus in this project is on RNA. So understanding non-additive changes in transcription that occur in these crossbred animals compared to their purebred parents. And then again, trying to map that non-additive variation in gene expression back to places in the nuclear genome that are acting and mediating in that heterosis process. And so our big hypothesis here is that gene expression patterns are going to differ between purebred average and F1 crossbred offspring. And what we call this F1 misexpression, right? So when that first generation crosses offspring, or the offspring in the first generation cross has an expression value outside of what we would expect given the average of the parents, we call that F1 misexpression. And our second part of that hypothesis is that this misexpression is driven in part by genetic variation that alters transcription in a non-additive fashion. So in a sense, non-additive EQTLs that are affecting this expression phenotype in a non-additive way. So the tricky part, right, and the reason that nobody has done this far is that common garden experiments for our cattle are just not possible, right? Our friends in the maize breeding world can put everything together in a really nice, again, inbreds raised in the exact same environment as their F1 offspring, replicates across environments, right, and really drill down into this. But for cows, unfortunately, we don't have the liberty of doing common garden experiments. So we had to find a system that gets as close to this as possible. Again, the U.S. Department of Agriculture also doesn't love funding these sort of super long-term population development projects. So we needed something that was relatively quick, that was controlled. Gene expression is a notoriously messy phenotype. And we needed something that we could generate a pretty large and powerful data set from. So what we opted for is to use an in vitro embryo system to start studying this heterosis of gene expression. And so the way that we did this the reason that we did this is for a couple of different reasons, right? First off, we can make as consistent an environment as possible when we're developing these embryos in a culture system, right? We're able to develop our F1 embryos right along embryos in the same environment. So that was really appealing. Try and pull as much of that environmental variation and gene expression out as possible. And then we also understand that the point where we're studying these embryos, which is a late blastocyst, so right before that blastocyst would hatch and undergo the implantation process, is when we squish them up, extract RNA and sequence. So the beauty there, of course, is that transition from maternally persistent transcripts, so the transcripts that were in that oocyte from an individual's dam, those have mostly given way to the embryo actually expressing its own genome, right? So we're actually seeing the expression that we want to measure in those F1 or purebred offsprings, rather than early on in this process where we'd likely be picking up these more biased maternal transcripts. So again, we think a nice little system for studying heterosis in a really clean sort of way. And so the three big questions that we wanted to ask in this project are what are the macro scale gene expression differences that accompany heterosis? So not so much digging into biology in this first part, but really are there big picture trends that we see, right? Is overexpression compared to purebred parents more or less frequent in those F1 offspring? Are there differences in the variation of genes expressed in the F1 offspring compared to those purebred parent populations? So we're interested in understanding macro as well as what the biological differences look like that underlie these differences between crossbred and purebred embryos. And then finally, trying to dig into that functional genetic architecture, map non-additive EQTL back to those genomes and understand how that might be modulating heterosis, not just at this scale of gene expression, but downstream across time scales and at different points in an animal's life. And so the experimental design that Garrett set up for this project was to effectively make a diallele cross between Angus and Brahman. So we've got a lot of divergence between these two populations, somewhere in the neighborhood of 250,000 plus years diverged. The Bos Indicus and Bos Taurus lines are. So lots of divergence should generate us the maximum amount of heterosis. We want to be able to observe heterosis if it's there. And so again, instead of trying to go through and do a large number of OPUs or something on cows, we were actually able to just follow animals of known genetic origin through the slaughterhouse, pull their ovaries out. So 24 Angus cows, 36 Brahman cows of known genetic origin, pull ovaries out, slice, get our oocytes, and fertilize with three different Angus and Brahman bulls in pools. And so what we were left with there, 63 purebred Angus animals, 100 purebred Brahman blastocysts, and then about 143 of our F1 individuals, and those are reciprocal in nature, right? Sired by both Brahman and by Angus bulls. So this gives us the ability not just to detect differences between these groups, but start to map parent of origin, breed of origin effects, in allele-specific expression at the level of the individual embryo. And so, again, at the end of the day, we're left with about 306 mature blastocysts. The other thing that we do to enable this expression QTL mapping study is we sequence each of our parents to about 10x. So we've got really reliable genotype calls on parents. We're able to go back and use the transcriptomes generated for individual embryos to impute those non-genic regions of the animal's genome, allowing us to undertake the EQTL mapping process using imputed genotypes here of very, very high quality because we've got this nice, neat little cross that we've made. And so just a quick little rundown on the breakdown of individual embryos, 163 purebreds, 140 crossbreds, so a pretty balanced set there. These are all sort of top grade embryos. We threw away embryos that weren't of sufficient quality. We really are only studying embryos. We wanted to remove as much variation from a quality standpoint as well. These are all late blastocysts, grade A embryos, exactly what we would be transferring to cows if we were going to do that, right? About 25 million reads per individual embryo. So a fair bit of sequencing depth there should allow us to detect some pretty modest shifts in gene expression if they're there. And so when we look across this data set to start with, a little bit curious. So our colors here going from purebred Angus in blue down to purebred Brahman in purple, but the largest driver of gene expression doesn't appear to be breed, which was counter to our expectations. We figured that subspecies would easily be the biggest driver of gene expression differences here. But it turns out that when we go through and look at these individual blastocysts, we see our male blastocysts clustering with one another and female blastocysts clustering down there and to the left. So nearly 30% of the variation in gene expression, even at this 100 to 150 cell stage, is being driven by sex differences in these embryos. That sex determination cascade won't happen for a long time, but even at a very, very early stage, the presence of that Y chromosome is driving some really massive shifts in gene expression in these two classes. And it turns out there's a whole bunch of differentially expressed genes between males and females. This was a little just a quick analysis that we've run and have a student who's actually digging into some more, digging into this data set in some more depth, trying to understand these differences in expression between male and female, some of which are very well known to be differentially expressed at this early stage, but many more that we're interested in understanding some of the biology here. But the long and short of it, though, is once we remove that sex effect, that batch effect due to sex, we see a nice separation between our purebred embryos in purple and blue, and then our F1 embryos cluster nicely in the middle between those two populations of purebreds. We find a handful of genes that are highly differentially expressed between our F1s and purebreds, but in general, a relatively small number of differentially expressed genes at these more moderate fold changes, right? So there are somewhere in the neighborhood of 2,800 significantly differentially expressed genes between purebreds and F1s, but very, very few of them would we classify as even moderately differentially expressed. These are very subtle changes between these groups. But again, when we start to dig into the biology, by far the most significant of all of the functional classes of genes that pop out in these analyses are cell cycle checkpoints. It appears that there's some sort of pretty substantial differences between our purebred individuals and our F1 individuals at these crucial cell cycle checkpoints, right? So trying to project that forward, I'd hate to go too far in any direction with this limited information, but again, it seems that this might potentially be affecting growth downstream, right? Some of these processes very, very early on in an individual's development could show up as heterosis in one way or another further on in these animals' lives, right? So some interesting biology as we continue to dig into these things. Some of the more macro level differences that we find, we test for differential variances just using F scores on a gene by gene basis here. We see that when we start to look at greater variance between, so we either put our F1 individuals together or purebred Brahman and Angus individuals into a bin here and ask what the variation is on a gene by gene basis. For about 600 genes in each classes, we see significantly different variances within our F1s compared to the variances when we pool those two purebred classes together. Of course, when we compare to within breed variances, nearly 10 times as many genes with significantly greater variances in F1s compared to either of the purebred parents, right? So obviously, in addition to average differences in expression, right? So differences in mean versus differences in variance, it looks like there's a little bit of both going on at the macro scale when we compare expression between our F1 and purebred animals. And the other trend that we start to tease apart is that in general, across all genes and across genes that are differentially expressed between our purebreds, we tend to see that underexpression in our F1s, so these density plots, the red line, are F1 individuals, we see that there tends to be a shift towards lower numbers of median scaled counts in our F1 individuals compared to our purebreds. And again, this F1 underexpression appears to be pretty pervasive, right? So it's not necessarily heterosis in the way that we would typically think in a phenotype where we get additional growth in that F1 individual. Actually, at the level of gene expression, it looks like heterosis is showing up as a value that's less than the average of what we would expect due to the two parents. And so we look at this dominance to additivity ratio as a measure of that. Looking here at different fold changes between our purebred individuals, right? So what we see is that the more highly differentially expressed a gene is, so these green and blue lines, these are highly differentially expressed genes between Brahman and Angus. When we look at them at the dominance additivity ratio of these genes, we see a much, much lower expression, right? So there tends to be more underexpression compared to overexpression in these genes that are highly differentially expressed between those two parent breeds. So again, these are big picture trends that we're continuing to dig into, but are excited about the potential of understanding this biology a little bit better. And then finally, as we start to dig into individual genes, we see every pattern that you can imagine, right? Where these are the sorts of evidence that we need before we continue to dig into functional trait or to functional mapping, right? EQTL analyses, where sometimes we'll see a breed biased activation of a gene, right? So here our F1s look about exactly like the average expression of a purebred Angus, whereas Brahman falls far below that. We call that taurine dominant activation. The same thing can happen with inhibition, where these guys, F1s look a whole lot more like a purebred Angus, but that is the lower of the two parents expression. We also see maternal driven, both on the indocene and taurine sides, right? Where you look a little bit more like your Brahman mother in some cases, or your Angus mother in some cases. And again, sometimes these are genes turning on when you have an Angus dam. So again, every pattern under the sun that you can imagine, we've got some pretty good examples of these non-additive gene expression cases happening. And so what we're working on right now is we've imputed these blastocyst genomes up to 10 or so million SNPs based on those parents that we sequenced to moderate coverage. We're in the process of going through and actually doing some variant mapping. So using various expression phenotypes, so whether that's in an additive or non-additive case, also looking at allele-specific expression, splice variants, and trying to understand the additive and non-additive components of these expression phenotypes, and ultimately drill down to the DNA variants that are associated with gene expression heterosis. And hopefully some other ongoing work is trying to map non-additive QTL across a number of phenotypes. So we're interested in understanding if even in this very, very early stage in development, we might be able to identify variants that are driving non-additive changes in gene expression that manifest later on in an individual's life in the heterosis that we're trying to capture across the beef industry when we crossbreed. So that was a really quick run through that project that has been a lot of fun. It's very basic. We're interested in understanding biology, but ultimately we hope that there's still a little bit of meat on the bone for us to continue to pursue maybe more relative or relevant phenotypes across our animals. So fertility, we've started doing some work on immune phenotyping and understanding this same intermediate molecular heterosis and how we might be able to predict that moving on into the future. So with that, I'm going to take a drink here and talk about the next project. This is led by a PhD student in my lab, Rua Muhammad. And Rua's question is, how might we be able to use omics for phenotyping? So really, this is a sort of a jealousy slide. I really wish that beef cows had MIR milk data like you have in the dairy industry. This is such a powerful tool, right? This is a routinely collected sample on individual animals. We can use this as an indicator for a number of traits, and I know that your groups have done that very successfully. So my question has always been, do we have something similar in beef cows? And we're never going to go out and milk beef cows with any regularity to get an MIR sample. But my question is, can we creatively utilize omics as a latent indicator for some of these complex traits in a similar way to how dairy would use something like a milk MIR sample. So that's what we set out to do was, understanding that phenotyping is really where there's a lot of room for us to continue driving genetic progress. So how do we measure new novel phenotypes? A lot of the work that my group does is interested in novel phenotypes. So it's sort of a cyclical thing, right? We figure out new, better ways to measure phenotypes. We generate genetic predictions. We accelerate genetic progress. If we can do that for traits that we want in the breeding objective, but that don't exist in a reliable way to capture a phenotype, that's, I think, where our future is pointing us as we continue to strive for genetic progress. So again, going back to this central dogma of biology, understanding that there are intermediaries between this DNA and the ultimate phenotypes that we're interested in collecting. We can use these to identify functional genetic variation and improve predictions. So we do a little bit of that in our group as well. But really, our interest in this project is, can we use these as proxy phenotypes for our hard to measure traits? And at the crux of all this is that it has to be cost effective. We can't go out and expect to measure population level phenotypes if a gene expression assay costs us $500, right? That's too big of a percentage of an animal for a producer to ever foot that bill. So are there ways of very, very cheaply and effectively measuring gene expression? And then can that be used downstream as a proxy phenotype for some of these things that we would really like to capture, but don't have the methods to do so? So our four main questions in this project are: which library or sequencing approach do we take? How deep do we need to sequence? And those are really two of the bigger ones, right? Those are going to be your major drivers of cost. So if we can get by with half the number of reads, but capture 95% of the information, that's a big path towards helping us reduce the cost to a point where this might actually be practical to do on a population level. I'm also very curious if we can use these reads from gene expression to sort of double dip, right? You both get a gene expression phenotype on an animal, and you're able to use reads from the sequencing to impute genotypes. So we wanted to understand how many variants we're able to call with these various methods, and whether or not those might be sufficient for us to do a reliable genotype imputation. And then ultimately, what's the all-in cost, and what are the paths towards driving our costs of these things down? And then I think this is yet to be answered. We've got a number of avenues that we're sort of looking at here, but ultimately, how do we make use of this data in practice, right? There's going to be some pretty substantial challenges in modeling, understanding how we use it as an indicator phenotype, how we maybe use this data in phenotypic predictions rather than just genetic predictions. So lots of questions there, but the first four parts here, this is funded by the Agricultural Genome to Phenome Initiative. This was a USDA funded project a couple years ago, and we're really excited about sort of what we come out of here with as being a nice way for us to start attacking some of these more population level challenges of gene expression phenotyping. So what we settle on is that one of our best ways to reduce the number of reads. So if we're going to do this on a lot of animals, we got to get things cheap. So we actually lean on this idea of 3' RNA sequencing. So instead of spreading evenly our number of reads across a transcript, we try and focus our reads on this 3' end of the transcript. Right. So we've got poly A and random primers, which is, again, it's not going to completely focus all of our reads to this a strict part of this gene, but it's going to very much shift most of the reads towards the 3' end. What this allows us to do is we're able to pick up smaller and smaller changes in gene expression with less and less reads because we're not having to share all those sequencing resources evenly across a full transcript. And so we've got better resolution to detect small gene expression differences. The thing we sacrifice here is we're not able to capture splicing and variation within the transcript. So if we're trying to call SNPs, we're only going to get it over here towards the 3' end of a transcript. But in our calculus, these are sacrifices that we're willing to make, and this is likely our only path forward for doing anything population level because of the number of reads that we can generate reliable gene expression phenotypes with. And so the workhorses of this, of course, have been automation and microfluidics handling. If we were trying to do this at full cost and full volume, we'd still be a really long ways away on cost. But this robot in our lab and the ability to do 1/10 reactions allows us to spread the cost of a library across 10 samples, right? So that's a really quick way for us to drive costs down on the library preparation side. And then, of course, understanding the number of reads that we've got to contribute to each sample was another big element of this work. And so RUA had a little test set. This is actually from an acute heat stress study that we opportunistically grabbed some samples from. We tested a couple of different library preparations, targeted about 20 million single end 150 base pair reads per sample. So we think that that's probably more than enough reads to detect these gene expression differences. And we get about exactly the same number of reads across both of these library preps. And then we did a number of comparisons, both between the two library prep methods and across a varying number. So we do a serial downsampling and test how many reads give us reliable expression phenotypes on the other end. Again, what Rua did is she said, if we want to study these aggregate phenotypes, right? These latent phenotypes, we need informative genes, right? Genes with a single read map to them aren't particularly informative. So she labeled these informative reads as those that have 10 reads per sample and that are expressed in at least half of our samples. So as we think about what makes up a reliable phenotype, this was our arbitrary cutoff. So as we talk about informative genes, that's what we're talking about. And what that looks like, this is just whole blood. So about 12,500 informative genes using this Takara library prep method. We lose about 1,500 when we move to the Lexogen method for library preparations. We detect many more differentially expressed genes in this Takara data set. This is sort of the theme throughout, is that it looks like a more reliable library preparation method. But the big takeaway here is that as she did these serial downsampling and started to ask how many reads do we need to get a reliable gene expression phenotype, what she sort of settled on is that regardless of what she used as the criteria, so these are expressed genes, she used her informative genes, she used her differentially expressed genes, it looks like somewhere between 5 and 7 million reads is enough to pick up almost all of the gene expression variation within these data sets, right? So these are 10 times serial downsampling. She downsampled at 500,000 reads, 1 million reads, 2 million reads, 5, 7.5, 10, and 12.5. And really that plateau looks like it starts to level out there right in the 5 million to about 7 million reads area. So again, a quarter of the total reads that we generate here look like they're giving us the vast majority of variation in expression. What we don't get, right, is that the more reads that we continue to contribute, we don't see any sort of plateauing in the number of variants that she calls in these files. So again, I think that this is sort of in line with what we'd expect. The deeper you sequence, the more variants you're able to detect, even when you're restricting your sequencing to these more fixed areas of the genome. But what she does find is that Takara, the SNPs that we call from those libraries are of much, much higher quality than what we get from the Lexogen approach. So these are really strictly filtered genotypes, 144,000 of them that she calls that are of high quality show up in known variant databases for cattle. So for me, that looks like likely enough SNPs for us to do a somewhat reasonable genotype imputation, particularly as we think about trying to use this for EQTL mapping. We should very reliably, with that number of SNPs, impute the flanking DNA sequence around genes with this sort of data. And so the cost part, of course, is what we're really excited about. So what we figured is that, and this is all internal cost for us, but we're in the neighborhood of about $20 per sample to get a gene expression phenotype on an individual animal. So this is really, really exciting. A quick little dirty lab spin down and into a Trizol reagent is all that we've got to do on farm to make these happen. We've gotten quite good at this. We're coming up on 2,000 or so probably that we've done so far. This is, I think, a really reliable strategy for us as we head forward to generate gene expression data on large, large numbers of animals very, very cheaply. So excited about this, and all of Rua's work on this project helped us narrow down sort of what the protocol looks like, focused on those Takara libraries, splitting those into tenth reactions, and sequencing somewhere in the 5 to 7 million reads per sample realm. All right. I think I'm good on time here. Enough to cover this last little bit. I don't have a ton of data, but this is really where the rubber hits the road as we think about applying that omics-based phenotyping to large groups of animals to try and understand complex phenotypes. And so the challenge, of course, and if you ask any producer or breed association in the beef world, if there is one trait that they would like to be able to capture that they can't right now, it is what is a cow's feed efficiency on grass. At the moment, right, we just don't have a good method for capturing that. We can feed cows a forage-based diet in a sort of feed intake bunk, right? And that helps us understand, is the forage-based feed efficiency different from feed efficiency getting a concentrated ration? That's interesting, of course, but there are so many other moving pieces that make a cow an effective grazer and converter of forage into product output, right? So really our interest is in trying to understand these metabolism differences in individual animals as they're grazing, as they're sort of out in their working clothes doing their thing. How do we identify cows that are more or less metabolically efficient from others? And also understanding that there's a big behavioral aspect to this, right? So again, I think that the phenotype of a cow forage intake in a more controlled setting, again, eating forage from a bunk versus a cow who's got to be out in the elements, especially as we get into more stressful environments down here in the southeast and southwestern United States. These are stressful places for cows to graze. Their adaptation, their adaptability to that climate is going to be a major driver in their grazing behaviors and how that manifests at the end of the day. And of course, differences in seasonal, regional forage quality variation, that's going to drive changes in an individual's rumen microbiome. That's going to affect palatability, some cows' preference, right? This is a really, really complex picture. And of course, those animals, their susceptibility to extreme changes in climate and environment only becoming more and more important. So our interest here is, can we find novel measures of cow efficiency that would enable genetic selection? And we're interested in applying some of these omics technologies in novel ways to help us fill in some of the missing pieces in phenotyping. The workhorse for us, similar to your groups there, we're also interested in understanding enteric methane emissions in these more extensive grazing situations. So the green feed pasture system is I guess our best tool for that. Occasionally a pain in the butt, but again, the gold standard for capturing these phenotypes on grazing animals, and the ability to capture not only methane, but carbon dioxide, oxygen, and hydrogen, can allow us to do some indirect calorimetry, understanding metabolic fluxes in these animals, and using those as proxy stand-in phenotypes for cow efficiency. We're also interested in understanding how we might be able to apply wearable sensors to estimate things like grazing time. That's something that we can sort of back ourselves into pasture-based dry matter intake if we understand how much a cow eats. So we've partnered with an Australian company, Cirrus Tag, that develops these little ear tag based sensors. They've got an accelerometer on board that estimate grazing time. They're solar powered and they're satellite connected, which again, something that in the dairy world you might not think a ton about, but anytime I'm on a beef operation, the thought of having a clear line of sight to a router or a box to process data from an ear tag based sensor, there are very, very few clear lines of sight, even on our research farms in the middle part of Tennessee. So the ability to have these things interface via satellite, be off the grid, solar powered, good for 5 years or so. These are a really attractive tool for us in starting to understand these things. So they've been fun to play around with, and we've just expanded the population that we've put these in recently, giving us some pretty reliable estimates, we think, of grazing time and other activities that these animals are doing, that we can help use to supplement some of these more fine scale, metabolic differences, or omics based latent phenotypes that we're trying to understand these cow efficiency phenotypes with. So again, this is a combination of precision phenotyping, real-time weight, using methane emissions and carbon dioxide and oxygen fluxes from our green feeds, as well as water intake. These activity sensors have been very useful. And then trying to tie these things back to molecular phenotypes. So can we find gene expression patterns, right? Using some machine learning to understand how certain gene expression patterns might actually be able to represent some of these phenotypes that we wouldn't be able to capture on an animal, or that we'd have to ask a producer to invest an enormous amount in to make that happen. We're also very interested in the microbiome, and in particular, how we might be able to capture variations in the microbiome without the invasive nature of having to do an orogastric tubing on an animal. So we've been playing around with buccal swabbing, which we think is an exciting way to not only get a proxy for that regurgitated microbiome that's in the oral cavity of these animals, but also use the reads from that to generate our genotypes. So it might be a case where an oral swab on an animal could stand in for a tissue punch. And in the process, you also get an informative proxy for the animal's microbiome that you could model alongside their genotype as you try and predict a phenotype. So that's a project that we've been working on with a company called GenCove, and is still very much in the works, but we're excited about the potential to utilize this microbiome in doing more phenotypic prediction of animals' methane emission phenotypes as well as feed efficiency. And so we've been on the ground for a little over a year, collecting samples. So we'll have sort of serially sampled animals from across the year, trying to understand how repeatable these gene expression phenotypes, these microbiome phenotypes are. And then overlaying those with seasonal variation in pasture quality, forage availability, and ultimately these multiple spot samples on our green feeds and real-time weights, understanding and growing heifers first. And then this year we've moved on to one of our larger mature cow herds, running a very similar passive data collection scheme. So excited about all this, and hopefully you'll have me back in a year or two, and we can talk about the big findings of all this work when we've put the pieces all together in our further down the line here. Our group is working on a number of other things that I didn't have time to talk about. Like I said, trying to expand this nice, neat, common garden understanding of heterosis in cattle into some real phenotypes that are economically important. So we're working with some folks in our vet school now on novel approaches to immune phenotyping, and then trying to apply this heterosis question there. I'm excited about that, and hopefully we'll have more to share very soon on that front. We also do some more traditional animal breeding stuff, right? So we've been very interested in fertility traits, novel fertility traits that we can extract from inventory-based reporting schemes. The beef industry is pretty woeful in their collection of breeding phenotypes. So we've got to back ourselves into breeding phenotypes based on calving records. Another thing that I'm envious of the dairy world for. But again, we've made some progress on some big commercial data sets here, and are finding some interesting relationships between phenotypes. The heifer pregnancy phenotype that's really the standard across most breeds doesn't actually explain much variation genetically in future fertility successes as a cow, right? That first rebreeding is really challenging, and that's a heritable trait that's quite different from a heifer getting pregnant. So trying to understand those alongside some of our partners at breed associations, and hopefully roll some of those tools out in the near future. We're also still very interested in imputation, particularly using low pass sequencing for imputation. We're working on optimizing imputation pipelines and strategizing how you allocate sequencing resources to make sure that the reference is adequately representative for the animals that you're trying to impute. And then various other things with gene expression, microbiome, and then recently have expanded some of our programs into the global south, working with the folks at ILRI on developing breeding and data collection programs in places where the red meat value chain is underserved, but very, very important to people's livelihoods. So with that was a whole bunch. I would be more than happy to answer some questions if you've got them. A big thanks to our crew, a part of them pictured there, our funding sources and research partners. But again, really, really excited to get to share some of this with you all, and would be happy to answer any questions that you've got.
Okay, thank you so much, Troy. That was a wonderful presentation. We're going to open the floor now to questions, either from online or from in the room. I see we have a hand up online. Carolyn, you're welcome to ask your question.
Yeah, sure. Thanks, Troy. It was a really great presentation. Really like all the stuff that you guys are working on. I've got just two questions.
Sure.
The first one is about the looking at gene expression differences for when you're looking for heterosis
Sure.
patterns. When you're looking at gene expression in those embryos, do you try to differentiate between like differences in state, like a subtle differences in stage of development of the embryo versus like a difference in the pattern of Brahman versus Angus versus the heterozygotes?
Right. So we do fit an embryo. We fit both stage and grade in our statistical model there. So we're hopefully as close to comparing apples to apples there as possible. So we try and pull that variation. There will be subtle differences in stage though I guess as an aside, there wasn't any difference in developmental rate between our purebreds and our F1 individuals. That was one thing that I sort of thought we might find, was that those F1 embryos were just developing a little faster, or they were a little bit higher quality on average. But it's pretty well a wash on those two metrics. But we do try our best to account for those in our models, so that we're actually looking at differences between those two different embryo types rather than quality or stage.
Okay, thanks a lot. The second question is just really simple. Like, we also put a lot of gadgets on cattle here as well. I'm in Alberta, Canada. I'm with Agriculture and Agri-Food Canada, but also the University of Alberta. So we tried those series tags on cattle probably about 2 or 3 years ago.
Okay.
And we just didn't have very good success with getting data points. So I was wondering, like, how many data points would you get over a day, for example?
Yeah, so I think that is at this point a little bit more, I would say it's a more practical producer tool than a research tool, because just from a power standpoint, because they're solar powered, they can only spend so much energy shooting stuff up to the satellite. So they'll process on board I think it's every 6 hours, and you'll get 4 data packets shot to the satellite a day. We're working with them right now on some approaches to basically offloading data at more frequent intervals to a Bluetooth station that's maybe by a water, that gives us a little bit more fine scale data that we need from a research standpoint. But from a producer standpoint, I think a lot of their goal is to have something that's as long lasting, off-grid capable, and a producer just needs that idea of what happened in the last 4 hours, and where the critter's at. So I guess there are certainly better research tools out there. We're interested in trying to understand how we can interface with them and help on algorithm development, and make them a little bit more of a high return on investment for producers in addition to what they do functionally now.
Okay, great. Thanks very much.
You bet. Thanks.
Thanks, Carolyn. Colin, if you want to ask your question, you can go ahead.
Can you hear me there?
Yep.
Yeah.
Hey, Troy, good to see you again. Congratulations on all the great work. I did have a question, and I must say, once we start talking about gene expression, I'm definitely outside my comfort zone. So apologies if this question is naive or anything by any sorts. But I did find it very interesting and correct me if I heard this wrong, that you've seen reduced gene expression in the crossbred lines.
Right.
And my thinking is that like across the genome wide, or just certain regions? Or because I'm thinking, is it only specifically in regions that have been fixated in Brahman and Angus, and the reason they've been fixated is because you want in those breeds for those genes to be overly expressed because they're somewhat economically importantly associated with other traits? Or kind of what is the reasoning possibly behind that?
Yeah, I think that the thing I've had to sort of change my thought process on that front, because at first I'm like, yeah, of course, like more transcript equals more protein equals more phenotype, but not necessarily, right? So there's certainly cases where a gene being expressed lowly, if it's say a transcriptional repressor or something, right? So it turns another gene off. If it's lower, right, then that means that gene gets expressed higher, right? So we're trying to think about these more in the context of gene networks too, right? Are there certain little hubs of genes that we know contribute to phenotypes, or in this case to developmental processes that are being shifted together in a different way in those F1 offspring compared to the purebreds? I guess the other thing too, right, is that we see more underexpression. So the F1s are underexpressed more in genes where there's a bigger divergence in expression between the parents. And so I think your question about, fixed alleles, right, or at least, divergent expression across parent breeds, I'd have to sort of hit the marker board for a second to think about how exactly that would work. But there's likely something there, right? Divergent selection across those two breeds, and the underexpression, when we make that comparison, is there's likely something there. But again, I'd have to give it a little bit more thought.
Okay, for sure. Thank you.
Yeah. Thanks, Colin.
Thanks, Colin. We'll go to Flavio now.
Hello, can you hear me, Troy?
Yeah.
Okay, good. I think you can see me now. Thanks. First of all, thank you so much for the presentation. Very interesting, and congratulations for all this work that you are doing there. Many projects, very impressive.
Thanks.
I have a question that relates to what Colin was asking. So kind of going to the basics, right? So if you read a book like Faulkner, right?
Mm-hmm.
And you like to understand what I call useful heterosis. That's
Mm-hmm.
the heterosis that the F1 would be better than any of the two parents, right? Because if it's not better, it's a little bit deviation from the average. It's not useful. It doesn't help.
Right.
So, but then if you look at this traditional quantitative work in experimental animals, flies, so there's two hypotheses that explains this useful heterosis, right? And if I recall one of course is overdominance,
Mm-hmm.
right? If you have an overdominance of one of the alleles, you could have a useful heterosis.
Right.
And the other one is epistatic interactions,
Right.
that you create in the F1 with the different alleles in different loci. So have you thought about this kind of trying to link what you are trying to look at the expression RNA back to the DNA, and with the original theories about why you have usable heterosis? Have you thought about that? Because I was trying to your presentation to try to find some ways to, maybe I need to dig further on the results, right? But I think you are going to be able to see or to understand what the quantitative experiments were showing, right? That you really need no additive effects, right? Either dominance, or in this case, over-dominance, or interactions between alleles from different loci. Just I like to ask you and see your thoughts about this.
Yeah. No, it's a great question. And from I guess from gene expression as a phenotype, and thinking about over-dominance, we only saw, I want to say, 2 or 3 genes that looked like they were truly over-dominant in our F1s compared to our purebreds, right? So phenotypically there, it looks like everything sort of skews not complete dominance, and rarely ever does that offspring exceed or fall below both of the parents, right? So that best parent heterosis doesn't look like it happens at the level of gene expression. I guess if I understood your question correctly too, we're trying to work our way backwards from to the DNA level now, right? So I think we'll have a little bit more again understanding as we do additive and non-additive EQTL mapping. I think that helps us understand a little bit more what the actual gene function is, right? Whether it's that it's not just those additive differences, right? But if we're able to map those large effect dominance EQTL, epistasis, of course, gets a little trickier to deal with at that level. Does that sort of answer your question, or did I miss something?
Yeah, no, it was not really a question. Right? It was just to use your brain. If you had think about like how will you connect to the old quantitative theories, right? What
Right.
you are looking now in the gene expression. That was my intent. I mean, really, you commented really well. I mean, and if you look even prior to knowledge of genes, or I think that the explanation was the animals will have a more biochemical flexibility to adapt to the environment,
Right.
and
Right.
therefore they will produce more, right? So it's just I was thinking about the old papers and what you are doing now.
Yep. Yep.
Thanks a lot for the comments, and thanks for the presentation.
Yeah. Thanks, Flavio.
Flavio, we'll go to Felico now.
Yeah, thank you so much, Troy. I mean, I want to follow everybody there. You're doing fantastic work there. It's great, and I know it's almost a one-man band there, so very well done. Just want to say a couple of things. We have, Christina and I and Flavio, we are working on this new large project, which there is the main focus, obviously, but there is a piece on the beef side where we are collaborating with Ireland and Australia, Sam Clark and Donna Berry, and also, of course, our own Canadian data. Will be great, I think, to have to collaborate with you, my friend, and to start that, because we also would like you to come to our meetings virtually. We always have it on Friday afternoon. Flavio can add you to the list and so on. So really thank you so much for. And by the way, Caroline is also leading another project which is on the same competition, on the forage. We just heard at the beginning of the question period. So there is a couple of projects that your work can be fit quite well as well.
Yeah, absolutely. No, would love to be involved in we're an open book as far as sharing data and stuff, right? Trying to I think your guys' track records at commercializing this and making the rubber meet the road. You've got a great infrastructure and team there. So if there's opportunities for us to share data and ideas and whatnot, we're 100% down to be involved. So yeah, if you'll loop me in, I would love to do that.
We will. Thanks.
We are a couple minutes over, but if anyone has any last questions that they want to ask Troy, either online or in the room. No? Okay, so we will let you go for today, Troy. Thank you so much for your presentation. It was very interesting and a great discussion. We will be posting this recording to YouTube, so we'll share that link with you. And we'll see everyone back here next week for another seminar. Thank you, everybody.
Awesome. Thanks, everybody.